We'll continue our discussion on metabolism with glycogen metabolism this time. And I know we've covered all the carbohydrate metabolism in the beginning, from glucose to pyruvate through the citric acid cycle and making ATP with the mitochondrial electron transport chain. And now we're going to go back and see what happens if we have an abundance of ATP and an abundance of glucose. We don't want to turn it into more ATP or even pyruvate or acetyl-CoA because that's irreversible. What do we do with all this excess glucose? And we're going to store it in a storage form of glucose known as glycogen. But as we generally do with these topics, we talk about degrading the molecules first, and then we'll reverse our talking of building it at the end. Okay, so I'll degrade the glycogen molecule with you first, then we'll discuss how we build it afterwards. Okay, so starting at the beginning, where is glycogen? What is it? Okay, so glycogen is a large polymer of glucose. Right? There are thousands of glucose molecules in this typical glycogen granule. A picture of one, or a depiction of one, shown at the bottom right here. We'll get to what the little yellow ball is in the center later, but all the other things you see on it, they look like little black sticks, but they're strings of glucose molecules hooked end to end. And all throughout this thing, there are a bunch of branching, where one glucose is now attached to 3 other glucoses instead of just 2. Okay? So what's the point of this glycogen? Well, it's our storage form of glucose. We don't continually eat, right? So we're not continually making food like plants would, or we're not continually consuming food like, you sit there drinking sugar all day. We tend to do other things. So this is a buffering system between meals, right? So it maintains your blood glucose concentration in between your inputs of more food. Okay? So it's rather easy to make glycogen, and it's rather easy to break it down. It's rapid. It's something that can be done very quickly. And unlike fatty acids, it can be used during anaerobic activity. Because if you degrade fatty acids, which we'll see in our next lecture, you cannot get glucose out of it. Right? You only get acetyl-CoAs. So here you get a glucose molecule, which we know we can get ATP from under anaerobic conditions by doing fermentation, just from glycolysis. And that's what's happening in your muscles. When you're doing a strenuous exercise, you need more glucose. Acetyl-CoA is not sufficient. Okay? So 2 major storage sites in the body, and every cell has a little bit of it, but we'll talk about it in our 2 major storage sites, which is going to be in the liver and in skeletal muscle. The liver is about 10% by mass glycogen, which is a large proportion. Now, if you think about it, 10% doesn't sound like a lot, but the most of the mass of the liver is in fact like the most of the mass of almost every cell. It's mostly water, right? And then after that, you've got a lot of protein, particularly in skeletal muscle. But then 10% of it is mass. 10% of the mass of the liver is a glycogen molecule, or glycogen granules. That's a large proportion for one molecule, or one type of molecule. And why does the liver need so much glycogen? Well, it doesn't need that much glucose itself, but the liver also serves to provide glucose for the rest of the body in between meals, right? So it maintains the blood glucose concentrations. As far as skeletal muscle, it's 2% by mass. It doesn't seem like it's much, but the skeletal muscle doesn't have as many jobs as the liver does. It's only acting as a muscle. So why does the muscle need so much? Well, it might take time for the liver to get that extra glucose down to the muscle cells if you're using them strenuously, so they want to store of their own. All right, so 2% by mass is in skeletal muscle. Of course, there's a lot more protein there. But again, mostly water again. So if I ask this question, which organ holds more glycogen in your body, there's 2 ways you can answer that. Do I mean which one holds more by unit mass? That would be the liver. It's 10% of its mass. Skeletal muscle is only 2%. Do I mean which one holds more overall in total mass, total amount of glycogen? Well, that would be skeletal muscle by far, because you clearly have more skeletal muscle in mass than you have liver. Right? Your skeletal muscle, unless you're wasting away and you're a very small child, your skeletal muscle constitutes by far more mass than your liver does. Okay? So where is this glycogen stored in these cells? It's in the cytoplasm. Right? So very easily accessible in the cytoplasm. Okay, so let's look at our glycogen molecule in higher resolution here. So at the top is a typical stretch of glycogen. It's not the entire molecule at all, but it shows all the salient features of the connections. So the second row of sugars, where there's 4 glucoses attached end to end, right, represents one of those black lines from the previous drawing, right? Just a straight chain of glucose hooked together. You notice every linkage in that whole row is an alpha 1,4 glycosidic bond. Right? We saw this before when we took 2 glucose molecules and made maltose. This is essentially a maltose that's been extended. So it becomes maltose. If I put a third one on, it's maltotriose. If I put 4, 5, 6, 7, I put a large number of them on, it becomes glycogen. The other difference here is we actually branch it. It isn't one long straight chain of glucose. We branched it. So the top row, again, is a bunch of 1,4 bonds, continuous, but connected to the second row by a new unique linkage. It's an alpha 1,6 bond. Okay, so here the alpha number 1 carbon, the OH number 1, is in the alpha position, as all of them are. And instead of attaching it to a number 4, which is always down for glucose, we've attached it to a number 6. Of course, number 6 is not chiral, so there's no need to mention whether it's alpha or beta. It's irrelevant, right, because it's neither. But it's an alpha 1,6 bond now. So now I have one sugar in my picture here that when I'm labeling the branch point, that sugar is connected to 3 other sugars. Every other sugar in this picture is connected to only 2, the one before it and the one after it. Right? So that one in the middle that's labeled branch point, that one sugar there, is connected to 3 other sugars. The one before it, the one after it, and the one above it. Right? So it's connected to 3 new sugars, that only one in there that's like that. So that's going to be considered our branch point. Okay? So to degrade this molecule, we need to do a few things to it. We can only degrade from what we call the non-reducing end. So if you think back to carbohydrates, carbon number 1 of glucose was the carbon that when it was in its open chain or straight chain form was an aldehyde. That's a reducing end. It could be oxidized, right? And then when it forms into its ring form, it's that hemiacetal formation. That's carbon number 1, and we said it could be alpha or beta. That's the end I'm talking about. That's the reducing end. The other end of the sugar, I don't mean number 6, I mean number 4 in this case, because they're all in their ring form. So the other end of the sugar is number 4, and it's a non-reducing end. It can't become an aldehyde at some point. It's part of a longer chain. So when I say we're attacking this thing from the non-reducing side or non-reducing end, I mean at the number 4 end, or in this diagram, the left side. Right? So this chain continues for a while, and we get to the last sugar in the chain. That's the one I can remove. I can't remove from the right side because there's other sugars here. Okay? And ultimately, these all condense into a single chain, and that's connected to that yellow sphere we saw in the center of the previous drawing, and that's a protein that's used to hold all this together. We'll talk about it more when we start building glycogen. So I need to release some glucose from the non-reducing end, and it's going to be released as glucose 1-phosphate. Now, you may immediately notice there's a severe lack of phosphates in this molecule. In fact, there's none. Right, so how am I going to get a glucose 1-phosphate out of this? We'll talk about that when we get to the enzyme. That enzyme can chop down those glucose monomers as glucose 1-phosphate off of this structure until it gets to a certain point. And then because of the arrangements of these branch points, this alpha 1,6 bond, the enzyme can't get any closer to the next bond to break. Right? And it's always a set distance away, and we'll talk about it. But then we're going to need to rearrange this molecule a little bit, or rearrange our glycogen a little so that our enzyme can continue. Okay? And then finally, all those glucose 1-phosphates that we've removed, we need to convert to glucose 6-phosphates, because that's a molecule in the glycolysis and gluconeogenic pathways. Right? So we can't use glucose 1-phosphate directly. We're going to have to convert it to glucose 6-phosphate. And as you're exercising, right, and you feel that exhaustion of I can't go on, and I can't, it's not as fast as it used to be, you slow down, you're running after a certain distance, a certain time, you've depleted your muscle of the available glycogen. That's what the runners call hitting the wall, right? It's the main cause of that first exhaustion. You may get your what they call second wind, and now the glucose is being delivered by blood from the liver. So you have more glucose available. But depletion of the muscle glycogen is that first feeling of exhaustion. You've run out of immediate sources of energy. Right, so there are 3 enzymes required to break this down. And the first one's the simplest one, although we're going to spend the most time on it. We're going to remove the sugar from the very end. right? From the very non-reducing end. So at the top here is almost the same drawing. I'm just looking at, if you can imagine, I'll go back for a second. If you can imagine any one of these chains, let's say that the middle chain here, all the way at the left, as far as it can go, there will be a final sugar where nothing is attached beyond it. That's what I'm showing here, the last two sugars. They're more connected, but I'm showing the last two of any one particular chain. And that's all this is showing. And I'm going to remove the very last sugar, right, the one on the left here. The enzyme that does this is called glycogen phosphorylase. Okay, so let's explain what a phosphorylase is. So a phosphorylase is an enzyme that breaks a bond using phosphate. And we've seen a similar enzyme. We've heard of hydrolases, right? We talked about those when we did our breakdown of peptide bonds using water, right? So our proteases like chymotrypsin, those were hydrolases. Those are also breaking a bond using a small molecule. In fact, all hydrolases do that. They break a single bond using a small molecule. Think of the small molecule as the saw you add, right? So, or the scissors or whatever you're adding, it's the thing you have to add to break the bond. In the case of hydrolases, that small molecule could be anything. It could be a water molecule, it could be a phosphate, it could be an ammonia molecule. There's lots of choices. So under the big umbrella class of hydrolases, there are subclasses. One of those subclasses is called a phosphorylase, which, as the name implies, it's the one that uses phosphate to break the bond. The vast majority of the hydrolases clearly use water. It's obvious by the name, right? But one of the smaller subclasses uses phosphate to break it. Another smaller subclass uses ammonia to break the bond, right? So it's a separate subclass of the big umbrella category called hydrolases. Okay, so we're looking at a phosphorylase here, which will break a single bond using phosphate. Now, think back to when you were doing organic, and you add water to a bond and it breaks it. You remember one side gets H, one side gets OH, and there were some rules on how it's broken apart that enzymes don't always follow. But that's how you broke the single bond, right? So you got one side got an H, one side got the rest of the molecule, the OH. If I were to do this with ammonia, one side would get H and one side would get an NH2. If I do this with phosphate, like in the drawing above, one side gets H and one side gets the remainder of the molecule, the PO3, right? And that's exactly what's happening. So we're breaking this bond, right here, and one side will get H, this O grabs an H from the phosphate, and this carbon gets the remainder of the breaking molecule, in this case a phosphate, right, gets the PO3. Right? So this is different from the other classes of enzymes we've talked about. So let's be careful and go through these three functional enzymes here, or classes or subclasses, to tell you what they do. For each of these, kinase, phosphatase, and phosphorylase, they have something in common. Any pair of them has something in common, and also any pair of them have something not in common. Right? So you can find 6 pairings here. So kinase and phosphatase have something in common. Kinase and phosphatase also have something not in common. And you can do that with all the other pairings as well. And so a kinase, as you remember, is from the category or class of transferase. It transfers something. Both phosphatases and phosphorylases are not transferases. They're in fact both hydrolases. Okay, so there's a difference right there. Kinases, you remember, transfer, in particular, phosphates. Phosphatases cut the phosphate off of something. They use water as the saw. And phosphorylases, they don't cut phosphates off. They use phosphate as the saw this time. It's playing a different role. It's not being cut off. It is the thing doing the cutting. So when you're done, it ends up on one of the products. So if I had to ask what do kinases and phosphorylases have in common, well, they end up with a phosphate on one of their products. And what do kinases and phosphatases have in common? They both start with a phosphate on one of their substrates. So there's lots of ways you can ask these questions about these 3 groups. Just know the differences among them. Okay? For our case here, we're going to use a phosphorylase, and in particular, we're using one called glycogen phosphorylase. We often drop the word glycogen from it because it's the most commonly discussed one. But if I call it glycogen phosphorylase, that's the name of this enzyme. And later I might just refer to it as phosphorylase. In the context, I'm talking about glycogen. Okay, so sometimes it's just called that. So how does it work? Well, we're going to add phosphate, orthophosphate, PI, or just inorganic phosphate floating around, to the glycogen granule. It's going to cut off that first sugar, or in essence the last sugar, right? And you end up with a glucose with a phosphate attached to it. So glucose 1-phosphate, Y on number 1, because that's the bond that was broken. And the remaining glycogen molecule is now 1 sugar shorter. It's the only thing that changed. Right? So this reaction is very specific. It's always giving you a glucose 1-phosphate. You can't get any other number there because that's not where it's attached. It'll always give you glucose 1-phosphate. And it will stop when it can't reach the next glycosidic bond. And it will stop 4 sugar residues away from any branching point. Right? So if we look at the next picture, at the top of the picture here, I have a glycogen molecule all colored for you again. The little hexagons represent glucose. And the number 1 will be on the right. Number 2, 3, 4 is the left where it's connected to the next one. And number 5 is the top left of the hexagon. The top right of the hexagon, is an oxygen, right? As you see in all the sugar drawings. And number 6 is not shown for most of the sugars except for 1 where we actually have it involved in something. Okay, so all these hexagons represent glucose. And my glycogen phosphorylase can come along from the left here and start breaking these bonds. Right? And I said I can break until I get within 4 residues of a branch point. Well, let's define a branch point again. In this molecule up here at the top, this one here, the yellow sugar that's attached to 3 other sugars, is the branch point. I can't get within 4 of that, which means I cannot remove the 4 yellow ones to the left of it, 1, 2, 3, 4, or the 4 on the other chain from it, the green and the 3 blues. So the only ones I can remove by glycogen phosphorylase initially are the 8 red ones here. Okay, 3 from this chain and 5 from this chain. I cannot reach this next blue to blue sugar bond. It's too close to the branch point. The enzyme just physically can't get close enough to the bond. Okay? But it's convenient that it's always 4 away. And when you're counting, you do not count the branch point. Think of that as 0. Then you count 1, 2, 3, 4. I can break the next bond, but no closer. Right? Same thing for the top. This is 0. I count 1. That's the green one. 2, 3, 4, the blue ones. I can break the next bond, but I can't get any closer than that. So that's shown in the diagram here. It's called phosphorylase in this diagram. Remember, it's glycogen phosphorylase we're talking about. It cleaves off those 8 red sugars as glucose 1-phosphates. Okay? So 8 inorganic phosphates come in, and I leave with 8 glucose 1-phosphates. That leaves what's called limit dextran. My phosphorylase cannot act upon this in its current state. So I need to do some remodeling, some rearranging. That's where our second enzyme comes in. Our second enzyme is called the alpha-1,6-glucosidase. Tells you what bond it's going to break. It's also known more commonly as the debranching enzyme because it's going to remove the branch point. Okay? And it does several things. And if you were to look at older texts or older lectures, say older than 2016, they're going to show this as 2 separate enzymes because we didn't know they were the same enzyme at the time. They're going to call one enzyme, which I'm not going to give you its name because it's confusing now, so don't think about it anymore. They're going to call this first step, where we move the 3 blue ones, by one enzyme name, and then we're going to call the second step the debranching enzyme. Well, the 2 groups that were studying these, in fact, were studying the exact same enzyme. It did both steps. So in 2016, we've affirmed that that's the case, and now we just know it's one single enzyme that does both steps. So we're not going to give it its old name, which was a terrible name to start with. And now we're going to call it the debranching enzyme. It does all of these parts. So what does it do? It looks for the shorter of the 2 chains, right? The 3 blue ones here are kind of in my way. I can't remove the green one because I don't have access to its bond. But if the 3 blues were not in my way, I could cut off the green one. So what this enzyme does is shifts a block of 3 glucose, right, from one end of a chain to another. So it moved these 3 from the green to the end of the yellow chain. It could have put it on any end available. I'm just showing one example. If there were 50 other ends, it could have put it on any one of those. But it just shifts it to another one. Okay? So that's essentially an isomerase feature, right? It's remodeling. It hasn't removed or added anything net. It's just remodeled it. Okay? So that enzyme originally was an isomerase, but it has another function now. After it moves those three, it also goes back and cleaves off the green glucose. Except this time when you cleave it off, it is not a phosphorylase. This is a regular old hydrolase that uses water. As shown in the diagram here, it uses water to break the bond. Of course, one side gets H, the other side gets OH, and that leaves you with just plain old glucose being removed. No phosphate added here. Right? So just H and OH. So it breaks this bond once more. It is a hydrolase, but it is not a phosphorylase. So the little question I wrote here, is this glucose phosphorylated? The answer is no. Right? So in our phosphorylase enzyme, when we cleaved off all these red ones, yes, it was a hydrolase, but more specifically, it was a phosphorylase. Right? So it cleaved them off as a glucose 1-phosphate. And then our debranching enzyme comes along and shifts these three, moves them to a new spot, giving the green one exposure, and then it cuts off the green sugar, the green glucose, the branch point glucose, right, the one that's causing the problem, into just plain old glucose and the rest of the chain. So I've removed the 1,6 branching bond. But I do not get a glucose 1-phosphate out of it. I only get plain old glucose. Okay? Once that's removed, you notice there's no limit of 4 anymore, right? My normal phosphorylase up here can come along and cut off these blue ones and some of the yellow ones now, right? How far can it go? Well, just like before, it can't get within 4 of something else that's large. This core here, which was that little yellow sphere I showed you in the very beginning, is actually a protein, and it can't get within 4 glucoses of the core. So you count 1, 2, 3, 4. It can break this bond, but it can't get any closer than that. Okay. And then lastly, we've created our 2 products. Basically, one is plain old glucose. You know what's going to happen to that. It's going to get phosphorylated by hexokinase right away. And glucose 1-phosphate. Well, that's not quite ready for glycolysis or gluconeogenesis, right? To release it as glucose if we wanted to, say in liver. But I need to convert this glucose 1-phosphate into an isomer of itself, glucose 6-phosphate. So we have an enzyme that does that. It's our third and final enzyme of this pathway. It's called phosphoglucomutase. So it's a mutase, which is, of the class isomerase, and it moves something bigger than a hydrogen. So a mutase is just an isomerase that moves something bigger than a hydrogen. We're clearly moving an entire phosphate here. We've seen something similar to this back in glycolysis, where we moved a phosphate in one of our final steps there. Except here it works a little differently. The enzyme that does this has a phosphate already attached to a serine here. Okay, so our glucose 1-phosphate has one on its number 1 carbon, and our enzyme has a serine that's been phosphorylated as well. Okay. When the sugar binds, the phosphate from the serine moves on to carbon number 6. Okay, so now I have a doubly phosphorylated glucose stuck in the active site of this enzyme. Right, before it leaves, it flips over 180 degrees. This sugar just flips around in the active site. It won't let it go because it's holding onto it very tightly. Flips it around and gives its phosphate that was on number 1 back to the serine. So here it is. Right, and then the glucose 1-phosphate or 6-phosphate can leave now. This is entirely reversible. If this thing were to not flip over and the serine were to give the phosphate directly back, the sugar would give it directly back to the serine, it would leave as glucose 1-phosphate. This is totally reversible. The delta G is essentially 0. Right? So how is it regulated? Is it regulated at all? I want you to think about this one. Do we want to regulate this enzyme? Do we want it on or off completely some other way? Or we would let the substrates and products control the direction? Which one do you think?
Could you repeat the question?
So this is a reversible isomerase, right? Like a lot of our isomerases we saw in other pathways. Do we want to regulate the activity of this thing? Or do we just let it happen and let it go in whatever direction the substrates and products dictate?
They probably regulate depending on what you need at the time.
Well, wouldn't the substrates and products do that? Right? If there's a ton of glucose 1-phosphate around, in other words, we're degrading a lot of glycogen, And there's very little glucose 6-phosphate around because we have no glucose available. So we're degrading our glycogen to supply glucose. This is clearly going to go in the right direction, or forward direction, to the right on the screen, right? Because there's lots more glucose 1-phosphate than glucose 6-phosphate. If there's an abundance of glucose, right? You ate a dozen jelly donuts and all the glucose influx into the cell. We don't want to degrade glycogen right now, right? So there won't be any glucose 1-phosphate around, but there'll be plenty of glucose 6-phosphate. So it'll store it as glycogen. So the substrates alone could regulate this thing. It's totally possible, and that's how we did a bunch of our enzymes in glycolysis as well. But could we regulate this another way? Is there a way to turn this enzyme off? Can you think of another way? This is not an easy question, I know. I'm just getting you to think about it.
You phosphorylate it?
I could dephosphorylate it, correct. And what would do that?
Glucose phosphorylase?
No, because you're not dephosphorylating a glucose.
Right.
We're dephosphorylating a protein. So what kind of enzyme would cut that phosphate off the serine?
Phosphatase?
A phosphatase, right. A protein phosphatase, not just a sugar phosphatase, right? It's protein phosphatase could cut it off. And if that serine is lacking a phosphate, then this enzyme cannot function, right? It can't do this isomerization. So that's one way to control it. There must be also what type of enzyme available to turn it back on.
A kinase or a phosphorylase?
Well, it'd be a kinase because I have to transfer a phosphate onto the serine, right? A phosphorylase wouldn't put a phosphate on the serine. It would cut a single bond using a phosphate, and there isn't a bond to break here to do that. So a kinase would put that phosphate back on, and that's like a lot of our regulation we've been talking about. You put a phosphate on something with a kinase, generally on a serine, to regulate it, but it's also involved in the mechanism here. Right? So it's a very easy regulation to think about, but it's also controlled by the flux of glucose with a phosphate on number 1 or 6 in either direction. And so if we look at two cell types here, liver versus muscle, in the glycogen in the muscle tends to stay phosphorylated. So if we degrade glycogen, we make a lot of glucose 1-phosphate from it, we do the isomerization over to glucose 6-phosphate, it tends to stay in the cell as glucose 6-phosphate. Because unlike liver cells, muscle cells, and almost every other cell, doesn't make glucose 6-phosphatase. Doesn't cut the phosphate off so it can export it. Generally, only liver does that. Right? And the liver would do that to export it into the blood to maintain the blood glucose concentration. Okay. All right, so let's look at how phosphorylase is regulated. Now, this is not the enzyme we were just talking about, not the mutase. This is the very first enzyme, number 1, that removes the sugars from the end. Every time I give this lecture, I always get looks at the end of class where the sentence you just said sounds like you just said phosphate 7 times and it makes no sense. Well, that's sort of what it sounds like too. So I'm going to go through this one step at a time. I've hopefully made the slides a little easier to understand this time. So phosphorylase, and I mean glycogen phosphorylase, that's the name of this enzyme. Remind me again what it does. What does glycogen phosphorylase do? I'll go back a few slides to help.
Remove sugar from the end?
Right, it removes the sugar from the non-reducing end, right? Cuts off those red ones above. That's exactly what it does. It uses phosphate as the saw to cut it. It is a phosphorylase, and its class is a hydrolase. Okay, so looking at this again, phosphorylase, the enzyme I'm talking about, accomplishes that chemistry. It starts to degrade glycogen. This enzyme, phosphorylase, exists in 2 forms. One we call phosphorylase A, and one we call phosphorylase B. The only difference in these 2 forms is phosphorylase A is phosphorylated. There's a phosphate on it. I know that sounds like I use the same word 5 times, but hopefully that makes sense. There's a little phosphate that's been added, right, to a serine, of course, as usual, somewhere on the enzyme. In fact, there's 2 of them, right? And phosphorylase B is lacking those phosphates. Okay? Both forms are still functional enzymes. They both do the degradation of glycogen. Both forms, phosphorylase A and phosphorylase B, are active, but phosphorylase A is more active. It's about up to 25% more active than the B form. Right, so I kind of drew that in the picture at the bottom with a little phosphate next to the A form, so you know it's more active. The B form can still do the chemistry. It's just not as active as before. Okay? Each of these forms, phosphorylase A and B, is determined by whether or not it has that phosphate attached to one of its serines. In fact, it's two serines. Okay? They also can exist in two other forms. This has nothing to do with the phosphates at all. This has to do with other things binding. Right? This is an allosteric regulation. Right? That means something is binding somewhere other than the active site, right? And modulating the activity of the enzyme. We talked about this during our enzyme lecture. So some things that can bind are AMP, ATP, glucose, glucose 6-phosphate. Right? We'll get to what those do in a minute. But when they bind, they can have the enzyme adopt one of two states. It's either in what's called the R state, or relaxed state, which is very active, or the T state, tense state, which is very, very low activity, right? Considered inactive, right? And what it does between these two is how it makes the enzyme more active or less active is it restricts access to the active site by glycogen, right? That's its substrate. So glycogen has to actually bind deep in the center of this thing, which is why it can't get any closer than 4, right? So it buries its tail deep in the center of this enzyme. Access to the active site, its tail takes a while to get in there. It's a very deep pocket. If it's in the T state, that deep pocket is very, very restrictive. It's difficult for it to get in there. Whereas in the R state, it's more relaxed. That's why it's called that. And the tunnel getting to the pocket is a little more open. It's easier for the glycogen chain to snake its way in there. Of course, if it's easier for the chain to get in there, that's its substrate, and it's more active, that means it has a higher affinity for its substrate, or a lower Km. It takes less substrate to get this thing going. Right? In the tense state, you can imagine it being tense, like contracting its alpha helices against each other. Then it has a tougher time for the substrate to get in there. So it has a lower affinity for the substrate, or a higher Km. We call it the inactive or tense state. That's where those names came from. And in fact, that's used in many other enzymes, this relaxed and tense nomenclature. And phosphorylase, this glycogen phosphorylase, was the very first one discovered that had allosteric regulation back in the 1940s. Right? So this is the prototype child of allosteric regulation. All the nomenclature comes from this guy. Okay, so we have two forms of regulating it here. We have a covalent modification, putting a phosphate on or not having it on, or an allosteric control. Some molecules binding, we'll talk about which ones bind which way in a minute, and it makes it have a different affinity for its substrate. It makes it easier or more difficult to bind its substrate. Okay? All right, going back to the first part, the phosphorylase A and B forms. Right? We know the A and B form is denoted by whether or not it's been phosphorylated. It's got a little phosphate stuck on it. Well, something must have put that phosphate on. That would be a kinase, correct? Kinases put phosphates on things. So the name of the enzyme would be something kinase. That's how we named them, right? If it's a kinase that puts a phosphate on a banana, we would call it a banana kinase, right? No such real thing. I'm just making up names for you. But if it put a phosphate on a dog, it'd be a dog kinase. It puts a phosphate on what? What is the thing being phosphorylated here? It's the entire enzyme, right? It's the entire enzyme called glycogen phosphorylase. We often just call it phosphorylase. So the enzyme that puts the phosphate on this thing is called phosphorylase kinase. It is a kinase. It's a transferase. It puts a phosphate on something. The something happens to be an enzyme with a name, phosphorylase. Is that making sense? I hope so far. I'm not confusing you. Right? Listen to it again if it doesn't make sense. What would take that phosphate off? Well, what takes phosphates off of things? Those would be phosphatases. So if I had a phosphate on a dog and I wanted to remove that phosphate, it would be a dog phosphatase. Here, I have a phosphate on an enzyme called phosphorylase, and I'd like to remove it. It would be a phosphorylase phosphatase. Now that sounds really confusing. So we actually don't call it that. It's just called protein phosphatase because it removes it from many different proteins, not just this one. Okay, so it's a little less confusing. Also, this thing is regulated by hormones from one cell to another, such as insulin, epinephrine, and glucagon. And we'll talk about how all three of these work. Okay? So to remind you on this slide, this kind of covers most of it. The phosphorylase, glycogen phosphorylase, can exist in two forms, A and B form. That's determined by whether it's phosphorylated or not, respectively. That phosphate, if it's in its A form, was put on by an enzyme, a kinase, called phosphorylase kinase. And if it's in its B form, the phosphate clearly had been removed by a protein phosphatase. Each of these forms, both A or B independently, can also exist in the R or T state. R being relaxed or more active, T being tense or less active, essentially inactive. And that's determined by allosteric regulation, other molecules binding, not covalently, but like AMP, ATP, glucose, and glucose 6-phosphate. And we'll see how those work in two different cell types. Okay, so let's talk about muscles first, right? And then we'll deal with the liver next. So muscle at the top. In muscle cells, we have glycogen phosphorylase, right? And its job, again, is to degrade glycogen. Right? It cuts off the last sugar, non-reducing unit. And in muscle cells, phosphorylase A, in other words, the phosphorylated version of phosphorylase, is always active. Right? If phosphorylase has been phosphorylated, so it's in its A form, it's always active, you can't turn it off. Right? It doesn't matter what kind of things are going on in the muscle cell, that one's always active. Phosphorylase B form in muscle, which is the glycogen phosphorylase lacking that phosphate, is only active if the cell needs ATP. How does the cell detect if it needs ATP? Well, you can't detect a lack of ATP. You can't detect a lack of something. You must detect something else. So in this case, AMP signals that ATP is really low. So AMP binds to this thing allosterically, right? There's a nucleotide binding site shown in the purple over here, and it can either bind AMP or ATP. If it binds AMP, then it indicates that this thing should be more active, right? And because we're binding AMP, the cell is in a lack of ATP state, so we should turn it on. This makes sense. Phosphorylase B, which is the less active of the two, A is always active, B is less active of the two, so it binds AMP when that's the scenario, and it makes it more active. So we start degrading glycogen like the cell needs. Makes sense. We turn from the tense to the relaxed state. Okay. Shown on the right is a diagram exactly what that explained. So we have the tense and the relaxed state here. In the relaxed state, we have AMP bound in those nucleotide binding sites. And in the tense state, we have ATP bound there. Now, some glucose 6-phosphate can also bind there, but it's less important than the ATP binding in the nucleotide binding sites. And that's the part I want you to know. Okay? In liver, it's quite different. In liver, we have, again, two forms, the phosphorylated form A and the dephosphorylated form B. The dephosphorylated form B is very rare in liver. Right? It's almost always immediately phosphorylated to the A form because the liver is very rarely out of ATP. Right? It's ATP rich all the time, right? Because that's where all your food goes. Right? So liver phosphorylase B is converted to A almost all the time, and in particular in response to glucagon. Right? The kinase is upregulated, and we make a lot of it from B to A. So I want you to remember for liver that's mostly in the A form, and in the A form, it's phosphorylated, as you see the little phosphates on there, right, little P's, and it's sensitive to glucose, right? So glucose can bind in the glucose binding pocket, which is a separate pocket than all the others. And in the liver, equilibrium favors the relaxed state when you have low glucose. If you have lots of glucose around, like you just ate, and there's a ton of glucose going into the liver, it's going to favor the tense state because glucose will bind and slow the process of this enzyme down. Right? What's the point of all this? Well, liver is sensitive to not its own concentrations of ATP, like muscle cells are, but it's more worried about the rest of the body. So it's not so selfish as the muscle cells are. That muscle cell is only concerned with its own state of ATP versus AMP. So it's sensitive to that. Whereas a liver is more concerned with how much glucose is in the blood, which corresponds to how much glucose is in the liver. So it detects that and adjusts its degradation of glycogen based on, does the rest of the body need sugar right now? Right? So totally different mechanisms, but basically the same enzyme. They're just adjusted from relaxed to tense state, or A to B form, depending on if you have ATP present in muscles or glucose present in liver. So they're looking for different triggers to turn this enzyme on. Okay, so here's a summary of that pathway on the right. So let's try to go through this. So we have phosphorylase B on the right, in its less active form, the non-phosphorylated form, right? And then ATP will be used by some enzyme, a kinase, to put those phosphates on. Right? We called that enzyme a kinase because it transfers a phosphate from ATP to the enzyme phosphorylase, converting it from the B to the A form. That enzyme, being a kinase, I name what it does, what it kinases, if you want to use that verb, right? It's a phosphorylase kinase, much like we had a dog kinase, or if you put it on a banana, it's a banana kinase, right? So whatever you're choosing to put it on, you name that in the name. It's a phosphorylase kinase because that's what it's phosphorylating. Well, like a lot of things in metabolism, it's a cascade of events. It's not just one. So my phosphorylase kinase here is also active when it itself is phosphorylated. Right? You see the phosphates have been added to it. It's active only when phosphorylates are present, so something must have put those phosphates on there, and that's yet another kinase. This one you've heard of before. It's called protein kinase A. Right? When we talked about our cascade of events and cell signaling, we turned on one enzyme called protein kinase A, and I told you then it had many targets. One of those targets is phosphorylase kinase. Right? So protein kinase A phosphorylates phosphorylase kinase, which makes it active, and phosphorylates phosphorylase. I know that sounds like a long sentence of the same word many times, but let me say it again. So protein kinase A, being a kinase, does what kinases do. They phosphorylate things. Protein kinase A in particular phosphorylates a large array of things. One of its targets is the protein phosphorylase kinase. Okay? So it puts these phosphates on. Phosphorylase kinase, having been phosphorylated, is now active. And it does what kinases do. It phosphorylates something else. Its target is very specific. It's the enzyme phosphorylase, also known as glycogen phosphorylase. And it phosphorylates it, of course, putting those phosphates on, making it from the B form to the A form. And now phosphorylase A, in its active form, can degrade glycogen better than it could before. The other thing that activates phosphorylase kinase is the presence of calcium ions. Okay, so both of these, the phosphorylation by protein kinase A and the binding of calcium, make it fully active. Either one alone is only partially active. Okay? And if you don't have either, it's completely inactive. So we need both signals. We need hormones, right, or nerve impulses or muscle contractions or whatever it might be, to provide these two signals. You already know about how to activate protein kinase A. We did a whole lecture on how to get that signal done. You also know about releasing calcium, right? We talked about how you could get the adenylate cyclase to make cyclic AMP to activate protein kinase A. We also talked about the slide brought to you by the letter C. Remember that? We have the lipid by our phospholipase C degrading IP3, degrading and making IP3, going to the cytoplasm, to the endoplasmic reticulum or sarcoplasmic reticulum in muscles, releasing all this calcium. Right? So that's in our muscle contraction. So we'll talk about these hormones and how this works in a second, but we've already covered it in reality. And we partially activate this kinase, right, and fully activate it, right, by putting the phosphates on and the calcium on. Okay, so let's talk about those hormones and how those three work. Okay, so I want to remind you the picture on the bottom right. We've done this before. We have epinephrine or glucagon binding. It's two of our hormones. Both of those use a 7 transmembrane receptor, right? just like we saw before with a G protein, the caps come off, we make a GTP, replacing the GDP. It activates adenylate cyclase. Adenylate cyclase converts ATP to cyclic AMP. Cyclic AMP goes and binds the regulatory subunit of protein kinase A, releasing the catalytic subunit, and now we have an active protein kinase A. We've covered all that before. So the active protein kinase A, as we just saw in the previous slide, does what kinases do, phosphorylates something. One of its many targets is phosphorylase kinase. When it phosphorylates it, it becomes an active phosphorylase kinase, along with the calcium, making it even more active, and it converts phosphorylase, my glycogen phosphorylase, from its B to A form. So all that's said in words here, and the diagram just fills in what we left off from last time. So if you look at this diagram on the bottom right, all the way through protein kinase A, we had covered before with cell signaling. I'm just showing you one of protein kinase A's many targets. There'll be more coming up, but one of them is to activate phosphorylase kinase, which then activates phosphorylase. And it's a cascade of events, just like we did before, so there's amplification in this. So as soon as you get an epinephrine molecule binding to a muscle cell or glucagon to the liver cell, we release this cascade of events, so protein kinase A and the release of calcium, all activating it to turn on the degradation of glycogen. Why does that make sense? Well, when you release epinephrine, your body is saying, or adrenaline, same thing, your body is saying to your muscle cells, you don't know it yet, but you're about to consume all your ATP. Right? You need to run from the bear that's chasing you. I don't care how much ATP you have right now, you need to start degrading glycogen. You're going to need more very soon. That's what epinephrine does for you. Right? Glucagon is released by cells to tell the liver to send us more glucose. So in response to that, suddenly using up all your ATP and suddenly using up all that glucose, cells tell the liver the same thing, send us more glucose. So the same thing, degrade your glycogen. Right? So both of these have the same effect on cells, the degradation of glycogen. Insulin, however, has a different effect. Insulin indicates that there's glucose in the blood, come and get it. So what do cells do? Well, the insulin cascade uses that tyrosine receptor kinase, right? We talked about it during the signaling lecture. It also activates a few other things. It activates a protein phosphatase called protein phosphatase 1, because there's many of them. And what will a protein phosphatase do? It'll come over here and cut those phosphates off of these things, lessening their activity or deactivating them. It also releases a phosphodiesterase, which will turn cyclic AMP, which is a diester, into just AMP. It'll degrade it. That shuts down the signal. So insulin has the opposite effect of epinephrine and glucagon on these cells, and it tells the cell to stop degrading your glycogen because there's a lot of glucose here. We're about to import more. We don't need to degrade the ones we have. Right? So they have the opposite effects. Okay? And of course, now that we've degraded our glycogen, and let's see if you get an influx of glucose and insulin's around, what do we do to make more? Of course, we told ourselves to stop degrading it, but if there's all this glucose around, we should make more. Right? So that's my segue into glycogen synthesis. We can't just reverse the enzymes we were just using. Every one of those enzymes, except for the isomerase, was completely irreversible. Right? They were hydrolases. Hydrolases are generally irreversible. Right? The isomerase was fine, so we can get back to glucose 1-phosphate from glucose 6-phosphate. I'm not going to talk about that one. It should be obvious. So if you have an influx of glucose, we can turn it into glucose 6-phosphate and then into glucose 1-phosphate. You should be able to see that pathway rather easily. But from glucose 1-phosphate, I have to put this sugar, this glucose, onto a growing glycogen chain, not degrade it. And that's not going to be energetically favorable. So I have to find a way around it. And we're going to do it like we've done lots of other things. We're going to activate the molecule. Okay? And here's how we activate it. I take my glucose 1-phosphate, right, and a UTP. Right? This is a lot like ATP, except it's uridine instead of adenine. And so we've done this with lots of different nucleotides before, but this is the UTP's job now. ATP had other jobs, and GTP had other jobs, and CTP had other jobs. TTP has no other job. It's kind of useless. Right? It's only used for making DNA. So UTP's role, other than being a substrate for making RNA, RNA is to help me activate sugars. Okay, so UTP plus glucose 1-phosphate makes what we call UDP-glucose. Now I want you to track the phosphates with me. Glucose 1-phosphate shows up with 1, and we colored it red here, phosphate. UTP, obviously the T stands for 3, so we have 3 phosphates. 1 colored in blue, 2 colored black. When I put these together, I'm calling it UDP-glucose. It sounds like the UTP lost the phosphate and it's connected to a glucose now, but that's not what happened. I lost both black phosphates, right? The ones colored in this picture. So the red phosphate on the glucose is still there, and the blue phosphate on the uridine is still there, the alpha phosphate, but the latter two, the beta and gamma phosphates, are lost as pyrophosphate. Where have we seen something like this before?
DNA synthesis.
DNA synthesis, or more specifically, RNA synthesis, right? It's actually more analogous because we don't have a deoxy here. Where else have we seen it? Or perhaps we haven't seen it yet, because I think it's our next lecture, sorry. We're also going to see it in lipid activation. I think that's our next lecture, sorry. But we're going to see the same theme throughout the whole thing. The example you talked about in DNA or RNA synthesis, we lost the latter two phosphates. That was a totally reversible reaction, just like this one is. What drives it forward? Well, we have that little molecule, pyrophosphate, at the end, and I can easily degrade it. It's degraded by an enzyme called pyrophosphatase, and it degrades it into 2 orthophosphates, exactly like our RNA synthesis and our DNA synthesis. Okay, so if I degrade that product, Le Chatelier's principle tells me this will go forward. I can't get that product back because degrading it is an irreversible reaction. So that irreversible subsequent step, it occurs after this, drives this reversible reaction only in one direction. So I make this molecule called UDP-glucose, where the total number of phosphates in this reaction is 4. 1 from the glucose 1-phosphate, 3 from the UTP. There's still 2 remaining. I could have called it UMP-glucose phosphate, but we grouped the phosphates together. That first name would have made more sense to tell you where they came from, but we call it UDP-phosphate or UDP-glucose because that's kind of how it looks, and 2 phosphates are leaving as pyrophosphate. Okay. The name of this enzyme is UDP-glucose pyrophosphorylase. So it's actually named for the reverse reaction, which can happen if there's pyrophosphate around. So it is a pyrophosphorylase, which is a phosphorylase, which is hydrolase. Right? So that's a reversible one. So what it's saying in the enzyme name is pyrophosphate comes in and breaks the bond between these 2 phosphates. Right? Where one side gets H, essentially, and the other side gets the pyrophosphate, right? But we're not going to have that happen in a cell because we're going to drive it forward by the degradation of the pyrophosphate. It's just named in that direction. They could have named it something else. That's just what they named it. If you were to try to come up with a better name for it, totally fine with me. If you want to call it a UTP-glucose 1-phosphate synthetase, or a UDP-glucose synthetase, that's fine with me too. It's just not the name that it was assigned. As long as you know what it's doing, I'm more happy than you knowing its name. Okay. So up here again, I apologize for this glycogen molecule, the way they drew these oxygens. I corrected one on one of our former slides, but I didn't get around to these yet, and I'll correct them later at some point. But there's no extra carbons here. It's not what this means. So we have our UDP-glucose we just made, right, using that weirdly named enzyme from a UTP and a glucose 1-phosphate. And I want to use this to attach this glucose that I've activated onto my growing chain. So this is my glycogen chain one more time. It's very long, and this is the last sugar. So just like before, instead of cutting one off, I'm going to add one to it. So I have an excellent leaving group here in UDP. Right? A uridine with 2 phosphates is an excellent leaving group, right? So if this OH, right, were to leave with it, right, so this whole thing could leave, and I can attach this sugar to this sugar, right? So I can attach these 2 carbons to each other through a new glycosidic bond. UDP is an excellent leaving group, so it leaves. So I've made a new 1,4 1 here, 4 here, alpha 1,4 glycosidic bond. Right? And more specifically, we can call it a glucosidic bond because it's between two glucose molecules. You don't have to know that. It's just another name for it. Okay? But this again, just like I want to make a reference to our RNA or DNA synthesis, it requires a primer. Right? You can't just put this on another glucose. It has to have a long chain already there that this enzyme, glycogen synthase, recognizes and adds one more to. So much like our DNA synthesis and RNA synthesis, our glycogen synthase must have a a primer already there, and it adds to it. It can't start de novo. Okay? And the activated donor is our glucose attached to a UDP called UDP-glucose. This is again irreversible because the UDP is an excellent leaving group and it can't break this bond on its own. Okay? But what if I want to start a new one? I don't want to build onto one that already exists or that I've chopped down a little bit. I want to start a brand new glycogen molecule because I need more of them. Well, here that starts with that little yellow sphere I talked about at the very beginning. It's a protein, right? So it's a protein called glycogenin, right? It gets one of these weird names because it's grandfathered in, right? So glycogenin has the capacity to generate glycogen, which is a very weird way to say that, right? But it's a protein, it's an enzyme, and it puts the first glucose molecules on itself. Okay? In fact, there's a tyrosine residue that gets the very first glucose. And the way it does that is if it replaces this glycogen molecule here with a big protein called glycogenin, and there's a tyrosine sticking out, right, which mimics this glucose. And UDP-glucose is used once again to put this glucose on the tyrosine. And that's what this enzyme does. So glycogenin auto-catalyzes the attachment of the first glucose. It puts the phosphate on itself, right? So it puts a phosphate on itself, however you want to say that, right? And it puts it on the tyrosine. And it does that for about the first 4 to 8 glucoses, right? I don't mean glucoses number 4 through 8. I mean it puts numbers 1, 2, 3, 4, 5, 6, 7, and 8 on. It puts up to 8 glucoses on itself in that first chain. So if you look at the figure at the bottom, you could probably color in the first 8 of those circles and know that those are the glucoses that glycogenin could have put on itself. It most certainly put the first 4 on because glycogen synthase can't get any closer than 4, right? Like it says at the bottom, it can only add to chains that are 4 or more. So that kind of mimics the same thing for degradation. Fortunately, glycogenin can put on the first 4. In fact, it can reach so far out that it puts on up to 8. So who put on number 5? It could have been glycogenin. It could have been glycogen synthase. Who put on number 7? Again, could have been glycogenin putting it on itself. It could have been glycogen synthase. Who put on number 2? Most certainly glycogenin did, because glycogen synthase can't get that close. Who put on number 9? Most certainly not glycogenin. It can't reach that far, right? So we know where the barriers are. It has to put on the first 4. Then numbers 5, 6, 7, and 8, we don't really know who put them on. We have 2 possibilities. Glycogenin could have put it on itself, continuing the addition, or glycogen synthase could have taken over by that point. But by number 9, glycogen synthase is the only one that can reach it. Glycogenin can't reach that far away. Okay, so hopefully, if you want to color these in, like the first 4 definitely put on by the core, by glycogenin. Numbers 9 onward were definitely put on by glycogen synthase. And numbers 5 through 8 could have been either one of them. Okay, so let's see. If we keep doing that, all we're going to get is something like this. Here's our core. We put on the first 4 by glycogenin itself. We could have put on a few more up to number 8. And then number 9 was definitely put on by glycogen synthase. Right? But if we do only that and continue, we just get one long straight chain. Right? That's all we get. How do I get those branches we need in there? Well, we have a corresponding enzyme, which is very nice. It's called the branching enzyme, and that's its name. Right? So it's kind of nice. It corresponds to the debranching enzyme. They do opposite jobs. So it takes a long chain like this, and it grabs typically 7. We're going to call it 7 so you can remember a number. It's going to grab 7 on the end, so they're labeled in blue here, plucks them off the end, and reattaches it somewhere else along here, right? It's typically not on the first four. This diagram shows it on number 4, but it's typically not on the first 4, right? But it reattaches it somewhere else through an alpha 1,6 bond. So now I've branched it. Right? So this molecule, instead of being one long chain, is a little smaller. Right? It's more compact. It makes it more soluble. Right? But there is a downside to it. What's the downside of branching it? Anybody think of a possible drawback?
It requires a lot of energy.
Well, it requires no energy to branch it. This is a free transfer. It's a bad idea to call it a transfer. I know I hate that word for it because it's not a transferase. It's an isomerase, and more specifically, a mutase, right? But what's the downside of doing this mutation or rearrangement?
It would make the molecule more globular.
It does make it more globular. That makes it more soluble. That's not a drawback. That's a benefit. So what's the one drawback? Think back to degradation.
You have to rely on multiple enzymes to degrade it instead of one.
True, but one of them, when I debranch it, results in what? What's the product when I use the debranching enzyme?
It's glucose.
Just glucose, exactly. No phosphates.
Yeah.
Whereas if I had a long straight chain there, let's look at our previous one here. If say we stopped here with our long straight chain, and I degrade this back to just having the 4 on the end, right? I would get all glucose 1-phosphates, right? But with the branching in there now, one of those is going to come off as just glucose. So I'm essentially losing a phosphate. I'm down 1 ATP because I'm going to have to rephosphorylate it with hexokinase. So every time I branch it, I'm sacrificing a future ATP. Right? Do I get anything out of it? Sure. It makes it more soluble, it's more compact, it fits in the cell, all these benefits. But it does cost me a future ATP. Right? There are no phosphates in this glycogen at all. But when we degrade it, we can think forward that this is going to give me 1 fewer glucose 1-phosphates. It's just going to be a glucose. So yes, I'm going to lose a phosphate, a high energy phosphate by doing this, but it's worth it. And that's the idea. So this branching into a more compact, more soluble thing is a cost, but a very minor cost. It's just 1 ATP for every branch point. Now, in our glycogen granule, there are many, many branch points. So yes, overall it's a lot of ATP, but it's not a large percentage of the molecule's ATP because each of these glucose can give me quite a bit. Okay, so let's look at this overall glycogen synthesis one more time. How does it work? Well, we take a UDP-glucose, which we made, right, and we incorporate the glucose onto the growing chain. Right, so 1 ATP was used, or 1 ATP equivalent, it's actually a UTP. We're going to call it 1 ATP equivalent, is used to put this glucose onto the chain. If I were to degrade it later, won't I get 1 glucose 6-phosphate back? It don't cost me any ATP to do that, so sure. It costs me 1 to put it in, but when I get my molecule back, I don't get just plain old glucose, except for the branch point. I get a glucose 6-phosphate, so it's ready to go into glycolysis, right? So I don't have to spend my ATP for hexokinase. I've already spent it in the past to make glucose 6-phosphate, put it in glycogen for now, and when I get it back out of glycogen later, it's ready to continue glycolysis. It didn't cost me anything to store it, except when I branch it. Because when I branch it, it costs me an ATP, right? Because I have to, when I get my glucose liberated later, I have to phosphorylate it again. It costs me 1. So if I take a glucose 6-phosphate and go through all of the oxidative phosphorylation pathway, I get roughly 31 ATP out of it. Why 31 and not 32? Because I said glucose 6-phosphate, not glucose, right? So I get 31 ATP out of there, and it costs me about 2 ATP equivalents, right? Not 1, 2 ATP equivalents to store it. 1 to put it on in the first place, and another to, 2 to put it on in the first place to make the UDP, right? Remember, it becomes UMP when I'm done. I lost 2 phosphates, and I get only 1 back when I get it as glucose 6-phosphate. So is it efficient? Yes, it's about 94% efficient. If you take 2 out of 31, you only lose about 4 or 5%. Okay? So if we did not branch it, we would have a ton of these molecules, these straight chain ones. It would be 100% efficient, but storing these would be a problem. And that's one of our glycogen storage diseases. If you can't branch it, it's called Anderson's disease. Right? It's one of your glycogen storage diseases. And you can still make glycogen, but you can't make it as efficiently as if you could with branching it. I know it says 100% efficient, but you can't make as much. So your cells can't store as much reserves as if they could if you could branch it. And you can only store as much as you're allowed, so any excess is wasted. So if you're eating a dozen glazed donuts and you can't store as much sugar as you're supposed to, where's all the extra energy going to go? It's wasted. It's going to be turned into fats. Right? So it's 94% efficient is very good. Okay? How do we regulate glycogen synthase? Let's think about it first. And it's unusual regulation, but let's think about it first. We can only regulate it by protein kinase A. That's what I'm telling you from the start. So protein kinase A is the signal. Right? And what does protein kinase A do? It does what kinases do. It phosphorylates things. So if it were to phosphorylate glycogen synthase, right, I want that to be an off switch, because epinephrine and glucagon binding turn on protein kinase A, and if they're turning on protein kinase A, telling me to degrade my glycogen, I most certainly don't want to be building it at the same time, right, and wasting ATP, or in this case UTPs. Right, so protein kinase A phosphorylates glycogen synthase, and that turns it off. That's a very unusual regulation. It's backwards to what we're used to. Usually phosphorylating something turns it on. Here it turns it off. And we'll see why that had to make sense in this picture. So we have 2 points of regulation. In our glycogen degradation pathway, glycogen phosphorylase was the target. Right? It was the thing that degrades glycogen. We often called it just phosphorylase. In the glycogen building pathway, or the anabolic pathway, glycogen synthase was the regulatory enzyme. Okay, so we'll abbreviate those GP and GS. Okay, so if you get a signal like glucagon or epinephrine, and it does this whole cascade, which you're familiar with up to this point, right? So you know all this stuff down to protein kinase A being activated. Let's do this one more time. Protein kinase A does what kinases do and phosphorylates things. In fact, it phosphorylates a number of things, 2 of which are described here. Okay? One of them we already talked about was phosphorylase kinase becomes an active phosphorylase kinase, which then does what kinases do, phosphorylates glycogen phosphorylase to become the A form, or more active, glycogen phosphorylase A, which then degrades glycogen. Okay, we did that already. Protein kinase A also does what kinases do. It phosphorylates glycogen synthase. And glycogen synthase goes from its A form to its B form. Again, B is less active. It does so with the phosphate this time. It's the reverse scenario from the kinase over here. So glycogen synthase gets turned off when it's phosphorylated. That's backwards of normal. And this had to be this way because one signal had to turn one on and the other off. If it turned both on, they would just be wasting UTP constantly, or ATP as well. So we want one on and one off by the same signal. So it had to be a backwards reciprocal regulation. How do we go the other direction? Well, we add insulin, for instance, right? It shuts this whole thing down. And then our protein phosphatase 1 comes along and removes all these phosphates. That turns my phosphorylase A into its B form, so less active, slow down the degradation of glycogen. My glycogen synthase goes from B to A, more active, starts synthesizing glycogen. And of course, phosphorylase kinase goes from being active to inactive, so it stops phosphorylating phosphorylase. So all these things had to be the opposite scenarios for one signal. Hopefully that makes sense putting it together. So an influx of glucose, what do you want to happen? I want to make glycogen, store it for later. I don't need it right now. I don't need to degrade my glycogen. I have glucose. An influx of, or a lack of ATP or a lack of glucose, well, we need to make more or send me more. So start degrading your glycogen if you're a liver cell, or start degrading it if you're a muscle cell, because I need to run, and stop making glycogen right now. So all these signals make sense if they had to be the opposite.